Hi, everyone. Good morning. We will continue what we left yesterday. We left yesterday at the formation of the nucleosomes. We will try to understand little more about the nucleosomes. Here I have written one nucleosome which appears to be a ball. That ball consisting of at least 9 different pieces of 9 moieties of histone proteins. Histone protein is a protein which contains lysine and arginine rich amino acids, and therefore they will be positively charged particles. Now if you look at this structure, the H3H4 proteins, they come together to form a U shape to begin with, and that means like this, H3, H4, and like that they come and form a U shape. To that they add other proteins, and ultimately there will be 8 proteins combined into form a complex, and that complex will be almost like, it's called the octamer. It is an octamer, it's a dimer. 2 levels of histone proteins will form a dimer, and totally there will be 8 of these units. And we have seen here the DNA molecule that is present in the cell will now get into and starts wrapping it over the histone complex, and they will have the first turn, and then they go all the way around, come back here and go to the second turn, and then they come out like this. It is coming out. This is going in. That means to wrap, they go in, they come up, ultimately wrap. And totally it is said that it is 1.75 turns will be there. It is not completely 2, but that will be 1.7. This is common to all the living cells. Whether it is any species of the animal does not matter. There will be two turns or 1.75 turns, and that will contain 200 base pairs. This is the common feature, underlying feature for all the living beings. How they are distributed: in the first turn there will be 73 base pairs, and in the second turn there will be 73 base pairs, and then the rest of the 54 base pairs will form what's called the linker DNA. Linker DNA, that's a linker DNA. The central one where it is wrapped on the histone protein, and that is called the core DNA. Core DNA. So all this together, that is the DNA histone complex that is formed, is one nucleosome. So like that, many nucleosomes may form in a cell. As I said earlier, nearly 3,000 million of those things will be there. One more speciality will be, in addition to these proteins, in order to protect the DNA molecule from the work of enzymes, there will be non-histone proteins wrapped up here and there. So that will be another kind of a wrapping takes place, so that the nucleases and other enzymes will not act upon that. When we look at the cell during the cell division condition, we find that some areas in the DNA will be loosely packed or they will be lightly stained. We can see the stain and also the structure, and that is called euchromatin. And there will be densely packed chromatin. That means the coils will be little more denser, and that is called heterochromatin. Why these things are, if you want to say, that would be very, very easy, because you should know all this packaging is taking place only to help and protect the genetic material that will go from parent to the offspring. Therefore, nothing should be lost in the process of packaging. Therefore, the nature take care of all these things. Probably in the heterochromatin area, where heterochromatin is possible, there perhaps DNA will start transcription. DNA will start copying. So the copying of the DNA to form its own copy duplication during mitosis, it has to be copied. Perhaps in the euchromatin area, they unwind like that, and then the copies are made. Perhaps that may be the reason why euchromatin is there. Heterochromatin is very tightly area that there will be no question of transcription here, because maybe each of the segments of the nucleosomes will have capacity to form some kind of a genetic material to be transferred into the cell. So therefore you say there will be 200 base pairs. What kind of sequence is there is not known. So that sequence may perhaps be transcribed to form the mRNA. So this is the total picture of the nucleosome. I just repeat now. You must know that nucleosomes are present in the are the units where DNA and the histone will form the complex. This happens in all the things, and ultimately this particular structure will keep on repeating itself, and final coiling may happen, and that coiling may look like that. A long thing like that, where you find here the histone packing will be here, histone packing will be here, and then like that you find a large number of thread-like structure, and this is what they call the bead on string model of the DNA packaging. So that is the first basic one. So this completes the work on the nucleosome. In the examination, if they ask you something about the diagram, try to remember this diagram and then please write it. As I said yesterday also, if you want to calculate the length of the particular DNA molecule, there is a formula, and that formula will be length of the DNA is equal to number of base pairs. We are looking at it, and that will be multiplied by the distance between two base pairs. That is the formula that is normally used in your textbook also. So NCERT textbook has given that particular thing. Kindly look into that one. And this comes to the end of the study of the nucleosome formation. Thank you.